We live in a world full of endless possibilities, with countless innovations by scientific minds across the planet aiming for humanity to flourish. In the field of medicine, diseases known to man are curable or incurable. Some of these diseases are due to defective genes, which are also referred to as genetic disorder. Through consistent research, we made the impossible happen because of genetic therapy. Good day everyone, we are Group 3, together with my groupmates, Ritz Igharas, Diana Paula Weves, Erica Mascolino, Mary Alison Galiana, Jasmine Saiki Gallo, Elra Flores, Art Micah Faith Garcia, and yours truly, Charis Jill Garcia. And this is our output for our subject, Science, Technology, and Society. Welcome to our informative vlog. In this video, we will be discussing about what is gene therapy. Do you want to know about gene therapy? Let's proceed to the next video.
What is gene therapy? Genetic therapy, also known as gene therapy, is an experimental treatment or prevention technique that uses genes. Some diseases are caused by errors or mutations in specific genes. Gene therapy delivers DNA into cells to replace mutated or missing genes or to introduce new genes. The particular intention of gene therapy is to treat a disease or to alter the body's ability to fight diseases. It does not correct 100% of human cells. As an alternative, every time a cell reproduces, it sends out a copy of a new healthy gene.
How does gene therapy work? With a gene therapy, doctors deliver a healthy copy of a gene to cells inside the body. This healthy gene may introduce a brand new gene and activate a damaged or mutated gene or take the place of an existing gene. These beneficial genes enter cells via carriers or vectors. The vector containing the genes will be directly injected into the patient when the gene therapy is utilized to alter cells within the body. Doctors take blood, bone marrow, or another tissue and separate out particular cell types in the lab when the gene therapy is used to alter cells outside the body.
In this video, we're going to discuss 4 of the strategies the gene therapy has: the gene augmentation, gene silencing, gene suicide, and gene editing. First, the gene augmentation. Diseases brought on by loss-of-function mutations which prohibit the gene from creating a functional product are treated by gene augmentation or enhancement treatment. This method of gene therapy involves introducing DNA into the cell that has a functional copy of the missing gene. It tries to produce a functioning product at levels high enough to replace the missing protein. Second is the gene silencing. Gene silencing therapy may be used to stop or silence the production of a defective gene when the insertion of a functional gene is ineffective in treating the disease phenotype. Just one faulty allele can induce the illness phenotype and any associated cell or organ failure in disorders that are dominantly inherited.
Third, we have gene suicide. The idea behind suicide gene therapy, also known as virus-directed enzyme prodrug treatment, is to use viruses as carriers to specifically transfer suicide genes into tumor cells. The specific enzymes that are produced by this gene can convert medication precursors that are not particularly harmful to cells into toxic byproducts in tumor cells, inhibiting their proliferation and eventually causing these cells to die. And lastly, genome editing, also known as gene editing, refers to a range of scientific techniques that enable the modification of an organism's DNA at specific sites at the genome. These technologies enable the addition, removal, or modification of genetic material. There are several methods for genome editing that have been developed. CRISPR-Cas9, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, and CRISPR-associated protein 9 is a well-known example.
What are the uses of gene therapy? Gene therapy aims to treat illness or strengthen your body's resistance to illness by replacing a damaged gene or adding a new gene. Numerous disorders, including cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS, may be treated by gene therapy. A few disorders, including severe combined immunological deficiency, hemophilia, and blindness caused by retinitis pigmentosa, have shown some success in clinical studies using gene therapy in humans. Ashanti De Silva's condition, which became the first gene therapy success story, is one particular example. She had experienced a string of debilitating infections by the time she was 2 years old. Adenosine deaminase, or ADA deficiency, was determined to be her particular kind of SCID. Her T-cell count was increased with regular injections of PEG-ADA, a synthetic version of the ADA enzyme, a treatment whose efficacy tends to decrease over time. Ashanti stopped reacting to the medication after receiving injection for 2 years. She would never become an adult without a radical shift in the way ADA was handled.
Common types of gene therapy. We have two types of gene therapy, and these are classified depending on the cells that are used. First is somatic cell gene therapy. A human gene is inserted into a living person's somatic cell. Somatic cells are cells that do not produce the eggs and sperm that give rise to the next generation. The goal of somatic cell gene therapy is to treat the patient's illness and not their offspring. Second is germline gene therapy. During this therapy, DNA is injected into the human body's reproductive cells, which can either be sperm or egg cells. The genetic variation of a person's reproductive cells will be corrected through the germline gene therapy, and this will be passed on to future generations. A genetic illness is permanently eradicated from a family line using this therapy.
So to summarize it all, we have learned what gene therapy is. It is simply a method for treating or curing diseases by changing a person's DNA. We didn't talk about how gene therapy works. One example of this is in replacing disease-causing gene with a healthy one. Third, we have enumerated 4 of the strategies that the gene therapy has: the gene augmentation, the gene silencing, the gene suicide, and the gene editing, which then bring us to the different usage of this gene therapy. We elaborated its usage on treating different illness and how it helped Ashanti De Silva's condition. We hope that this vlog has now given you ideas what a gene therapy is, along with its different purposes. Leave a reaction and comment below to leave any feedback. Thank you for watching.